be fully well recorded. Of course, we have recording from last year that don't change very much. But yeah, it's nice indeed if you can be live in the lecture room for this reason. Because so far, my lecture actually, the first one went wrong, and the second halfway, I think there is no audio. So yeah, it's not very useful. Yeah, but I've put already that one from last year, so you can look at that. Okay, let's start with the lecture of today. I want already to let you know it's going to be a bit kind of heavy in a way that we treat a topic that maybe is not for everyone, not everybody is very familiar with it. We will go much more into the molecular pathway. So stay with me. If you don't understand, please raise your hand, because it's easier to discuss when we are on the topic. And if it really takes too much time, I have time to stay after the lecture a bit longer if someone needs, okay? Okay, where we are now, we start week 2. So in week 1, we did more how the body reacts when we see food, right? When we look at food, when we taste food, and how it's processed inside our body. Week 2 until 4, they are much more focused on the cellular level. What happened then when we have big macromolecule, right? The most important are indeed the protein, lipid, and carbohydrates. What happened on the cellular level? And today, and especially, we will focus a bit more on ATP production. You did already ATP. You treated, today is Tuesday, yesterday with Evert, yeah? Again here, just for having a smaller link with the last topic of yesterday. So we have a recap. This is how the molecule of ATP look like. You don't have to know. You don't have to draw. At my time, which is not very long, but I had to know how to draw this structure. But basically, in ATP, as the name suggests, is adenine triphosphate. Yeah. So adenine is a structure that is the main element, let's say, of ATP. But the energy of the ATP molecule is actually in the binder of the triphosphate group to the adenine. So every time we break a phosphate, we liberate a certain amount of energy. And I put it here in the picture here in the table for you. And depend how many phosphate are left, then we talk about ATP with 3 phosphate, ADP with 2 phosphate, diphosphate, and monophosphate with only 1 phosphate. And you can imagine, because I just told you that the energy is in the bind of the phosphate, the less phosphate you have, the less energetic will be the molecule. So ATP is more energetic than ADP than AMP, right? If we do hydrolysis, so just put water with the ATP, what happens is that you can start to break down the molecule and make free the energy. Energy is measured in kilojoule per mole. So in this case, if you remove only 1 phosphate, you will have about minus 31 kilojoule. Sorry, kilojoule. Yeah. If you remove another one, it's going to be the same, of course, because the energy is in the bound. However, if we go from ATP directly to AMP, so with the molecule with only 1 phosphate, it's like that we break and we remove 2 phosphate group, right? And then what you obtain is AMP plus inorganic phosphate, which again with water. So it's a 2-step reaction, will liberate the 2 phosphate. And indeed, if you do the addition of these minus 38 and minus 24, you actually also arrive at minus 62, which is the total amount of energy. So why it is important for you to know this? Through the course, we will do sometimes, I will show you some reaction in which from 1 ATP, we don't make ADP. So it's not only that we liberate the amount of 1 phosphate energy, but we make AMP. And in that case, if that happens, if we go from ATP to AMP plus phosphate, then what we assume is that this is the equivalent of 2 ATP energy. Yeah? Is it clear for everybody? Because it's like basically that you do this reaction in step. Yeah? But you do as a single reaction. So the equivalent of energy would be 2 ATP. Yeah? Is it clear? Yeah? Okay. So you did already, I think, this a bit with Ever. So again, let's recap. This is the structure of glucose, right? The molecular structure. So if we burn glucose just in open air, basically, what we get is the carbon out as a molecule of carbon dioxide. We liberate water. So this is the chemical reaction. And we liberate 2,816 kilojoule per mole of energy. This is the energy content in glucose if you just burn in the open air, and this mostly release all as heat. However, in our body, in the cell, we don't do that. That would be really a big waste of energy to have all in heat. What we do, we entrap part of this 2,800 kilojoule in ATP molecule. Yeah? So how do we do that? Well, you will see by the end of this week that 1 mole of glucose in general lead to the formation of 30 mole of ATP. Yeah? And since each mole of ATP, so 1 ATP, is the equivalent of energy of 33 kilojoule per mole, it means that we are able in total to entrap 990 kilojoule of energy of glucose into ATP molecule. While the rest, so if you do 2,816 minus 990, which is in ATP, the rest then will be released in heat. And if we do the percentage of how much energy we can entrap in ATP molecule in the cell, it will be about 35%. And this is why the efficiency of the reaction of catabolism of glucose in cell in presence of oxygen is actually equal to 35%. Yeah? Then there are some aspects to keep in mind about ATP, because ATP is actually our currency in the body, right? This is how we exchange actually energy through different tissue, through different cells, inside the cells, depends where it's needed. So let's do a couple of exercises just to break a bit maybe the ice together. Yeah, you can also do together with each other, next to each other. If we consider an average body of about 70 kilograms, you should know that we can only store in this body about 3 millimole of ATP per every kilogram of body weight. This is the max that can be stored. And you should know that the molecular weight of ATP is 507.18 grams per mole. So I want you to calculate now how many grams and how many mole of ATP we can store in such a body of 70 kilo. Yeah, I leave you a bit of time. Of course, I have an answer later for you. Going also from gram to mole, this is basic lab skill calculation. So you will also need, for every thesis you will decide to do in any lab, yeah? Be careful also to the unit of measurement, right? I gave you millimole. I'm asking you mole of ATP that you want to store. And then gram by using a conversion with the molecular weight. Did anybody already answer the first question? Can anybody want to share? I need a number of mole. Who can tell me how many mole? Yeah? Very well. 0.21 mole. Yeah? And how many gram? You have it as well, I guess, or not? Yeah? Yeah, very well. So this is for the people that don't know how to do. This is very straightforward calculation. If you can keep 3 millimole of ATP per kilogram of body weight, then you multiply by that. You know the total amount of millimole of ATP because I give you millimole. And then you convert into mole, yeah, by divided by 1,000 because 1 mole is 1,000 millimole and the other way around. And then normally you multiply by the molecular weight to convert between molarity and grams. Yeah? So basically a body of 70 kilo can store only 106 grams in total of ATP. Yeah? Then you know that glucose This is the kilojoule of gram of an energy content. The molecular weight is 180 gram per mole. So let's say this body, let's assume that this body to live an entire day just on glucose. Let's put that this person, 70 kilo, can only drink glucose or can only eat glucose for 1 day. How much glucose is needed to cover just the basal requirement to survive the day, right? So just the basal metabolic rate, the maintenance requirement, which is 8.5 megajoule per day. So this is the total energy needed by this body, megajoule. So be careful again, yeah? You know that glucose is this energetic value, kilojoule per mole. So how many gram and how many mole it means this person should eat of glucose just to keep himself alive? Anybody want to share how to do? I don't need really the final number, but I just want to know how you think about it. Yeah, sure. Yes. Yes, very well. That's a really good one, indeed. So you have 8.5 megajoule. Again, always be careful also in the exam. Please be aware of the unit, because sometimes they are tricky, the answer. And you normally have only multiple choice questions, so be careful with that. So you have megajoule. That means 8,500 kilojoule, right? You divide it by the energetic content of glucose per gram. And what you obtain first is about half a kilo of glucose to eat, just to survive the day. And then you divide it again by the molecular weight, by the gram per mole. And then in this case, you obtain that you can eat. It's the same way of saying I eat 500 gram, 545 gram of glucose or 3 mole of glucose. Yeah? It's just a different way of expressing it. Now, if we look at the ATP, what does this mean? If you know that each mole of glucose, it's 30 ATP, what does it mean then? How much ATP we can make with this glucose? It's a very easy one, right? How much it is? Yes, yeah, it's 90 ATP. And if we convert now again in gram, yeah, it means that with that amount of sugar, we will get about 46 kilo of ATP. But here, of course, we have an issue, right? What's the problem now that show up? And how we solve it this? Who can answer this? What do you see? There is a big problem, right? What did I tell you at the beginning? How much we can store? Yeah? So this means, well, if we use all the glucose just to survive the day, we would produce 46 kilo. But where do we put all this ATP? What do you think of it? Okay, I will give you the answer. Yeah, please go ahead. You use it? Yeah. Part of it?
And you store the glucose first, I assume, and then the ATP is made throughout the day so that it doesn't all have to be stored at once?
So you think it's kind of given in small portions so that you always have? No, that's not the reason. It's also a big difference. So even if you store a bit as glycogen or as a fat or whatever, it still doesn't solve the problem of 100 gram compared to even 20 kilo. Anybody want to give a try?
Anybody want to give a try?
No?
Anybody
Okay.
want to give a try?
We use, yeah, go ahead. Sorry, I couldn't hear.
There's also the 35% efficiency
Yeah.
that we have, so many of it, like 65% goes to eat.
Yeah, so now we consider this already, right? Because I say for each mole of glucose, we can only do 30 ATP. That's already the 35% of efficiency. So all the rest is already lost as it. So we have a trick, of course, in our body, which is kind of a conversion of energy, right? We don't keep all the energy as a free pool of ATP. We make ATP indeed available when we want, but not by dosing the glucose that is catabolized, but by transferring energy with the help of different other molecules in our body. And these are the coenzymes, and I will talk about it now. Yeah? So conversion of energy, and this is one of the aim of today. I will talk about oxidation first. What is this process so that you can arrive to understand the conversion of energy. Then we will talk a bit about mitochondrial structure and function and uncoupling. This is really HAP expertise, so if you see me talking too much, please stop me. I might get very enthusiastic about it. And then the second part of the lecture is about the TCA cycle. Yeah? So this is what you will do in this week, I believe, and also half of the other one. So, but today, so the choice of effort, which I can imagine, why is that? We are actually starting from the end, the final product, which is the energy currency that I told you, ATP, right? Then we will go through oxidative phosphorylation, which is the process used to make the majority of ATP in the cell. We will talk about the hydrogen carrier that transported the energy in the cell. Then we will do the TCA cycle, also called Krebs cycle. So if you hear this, it's always the same cycle of TCA. And then we will arrive at acetyl-CoA. And you see that acetyl-CoA is a very central molecule for all the big macromolecules that are digested in our body. And then specifically, I think you will do carbohydrate, if I'm not wrong, with me and Evert. And then we do protein with me and fat again with Evert. So we go back to then the single pathway for each macromolecule. But we start from the common product. Yeah? So when we look at ATP, I told you that it's it comes from oxidation of nutrients, basically a kind of a breakdown of nutrients, right? ATP can be done in two different ways in the cell, through different processes. One is called direct, which is the substrate phosphorylation. In this case, you do ATP by using directly ADP. I showed you before, right? It's the ATP with 2-phosphate only. You add the phosphate, and then you make ATP. This reaction, however, requires a lot of energy. It really demands a lot of energy because to make the new bond, it takes a lot of energy. And there are few in our body, but this is not very common, actually, in the metabolism. What is most common is the indirect production of ATP, and this happens in the oxidative phosphorylation. And the main difference between these two is that one is anaerobic and the other is aerobic. Who can tell me what it is? That's nothing to do with the sport and gym, right? Aerobic. What it is? Anaerobic and aerobic. Without or with what? Oxygen. Yeah, just speak out. Yeah, you don't need to raise your hand. So when we look at the formation of the ATP by substrate level phosphorylation, and here I show you few reactions that actually happen in the glycolysis. You will see this with Evert next time. As I told you, it's a direct phosphorylation. So you have a substrate, which is ADP, which is a form of the ATP incomplete, kind of, right? And then what you do, you just need to add a phosphate from another molecule to this ATP so that you can obtain ATP. It's a very, very energetic reaction. How do you do that? In this case, for example, you convert that biphosphoglysarate in 3-phosphoglycerate. So you add the phosphate group from one molecule to another. This is the direct phosphorylation. Here is another example when we do from phosphoenol pyruvate to enol pyruvate and then eventually to pyruvate. The same. Same principle, yeah? You add on this, so you just transfer, but this transfer requires, first of all, a specific enzyme that has to work, that you can read here in blue. And normally these enzymes are always called kinase. When there is a direct phosphorylation, you normally have always a kinase working. And it actually doesn't really require oxygen to happen. Indeed, in the first part of the catabolism of glucose in glycolysis, you actually don't need oxygen, yeah? And it's with this special reaction I show you, they happen in the cytosol. But we will have also some in the mitochondria. So again, if we want to see the link then between ATP utilization and oxidation of metabolic fuels, and these are protein, lipid, and carbohydrate, right? What you have basically when you eat and these nutrients arrive in the cell, you just break down them completely. What we mean with breaking down in metabolism, it means that the complete structure of carbons will be released as CO2 molecule. The rest, the energy content, the gross energy of the molecule, will be partly lost as heat, but partly will be used to make ATP through the mitochondria deoxidate phosphorylation by using important molecules that are called coenzymes. Think about the coenzymes like a kind of messenger. They transfer energy from one molecule, and then they arrive specifically at the mitochondria where they release this energy. And that's why we have this cycle. So they can accept energy when they are in the oxidative phase. I will show you in a moment what it means. And when they have the energy, they are called reduced enzyme. And then they go in the mitochondria, specifically at the electron transport chain, release energy, get back in the oxidative form. We want these because we need them to actually collect the energy from the breaking down of the nutrient, right? And then in this case, through oxygen and formation of water, what you have, you also have a formation of ATP, yeah? And ATP can be used, again, for physical and chemical work when there is the need from the cell. So what is oxidation? Now I mentioned already a lot of time. Well, you can think of oxidation, like the name suggests, reaction with oxygen, right? So you have a glucose, I told you before, open air. What you obtain is just carbohydrate, water, and heat. A lot of energy as heat. However, you can see a lot of oxidation also in the campus when you go around and find this bike completely forgotten for years, maybe somewhere in forum. They are all rust, right? That's also oxidation. But that's a different oxidation. It has to do more with the iron as an element that actually goes from the 2+ form, which is the oxidative form to the, the reduced form to the oxidative form. Yeah? When we talk about metabolism, most of the reaction of oxidation, they are actually called, oh no, sorry, it's the next one. There is another one that is dehydrogenation, which is oxidation of alcohol to aldehyde. We have this also in the future. When you will look at this one, you will say, I know that. So in this case, you have an hydrogen atom that, as you know, contain a proton and an electron that will be actually carry the energy from the molecule. And this process of dehydrogenation is actually also what happens a lot in our metabolism. I will explain you in a moment a bit better. But basically, hydrogen, as you know, is the simplest element, right? Chemical element. Is made by a proton and electron, but in this is actually the electron that is highly energetic. Proton is just there to neutralize the negative charge so that the molecule is stable. You don't want to have a molecule with a negative sign around your body because normally they are very, very much reactive. And we will talk a bit in detail later on. And this is why we have, in this case, FADH2. I'm going to spoil, is already one of the coenzymes that actually carry energy to the mitochondria. And you have the oxidative form, which is this one. And the reduced form is always the form that contains the hydrogen, that actually contains the energy. And when you look at that, always look at hydrogen that move. Because I told you, although the energy is in the electron, there is always a transfer of hydrogen just to stabilize actually the negative charge. In this case, for example, you have glycerol triphosphate converting in dihydroxyacetone phosphate. And in this case, you get these two hydrogen transfer on the molecule, and you get a completely different molecule. This now is quite energetic. Yeah? And you will understand in a bit what I mean with that. So when you think about oxidation reaction, this type of oxidation reaction are always present in the human body as a part of redox process. So they are dehydrogenation reaction, but sometimes in short term we call them redox Redox is reduction and oxidation. So for something that is reduced, there is always another molecule that oxidated. Yeah? Do you understand this concept? Because you can imagine if a molecule loses electron, it gets oxidated. If it acquires electron, like hydrogen, then it's reduced. Yeah? And I have this comment, over and over in a few slides just to fix it. Because you can imagine if a molecule loses electron, it gets oxidated. If it acquires electron, like hydrogen, then it's reduced. Yeah? And I have this comment, over and over in a few slides just to fix it, let's say. Yeah? So in this case, for example, if you see oxygen plus 2 hydrogen, you have water. Then you have in green, the oxygen become the acceptor, the electron, of the electron, sorry. And then the substrate is actually the H here. Yeah? Just for you to know, there are some bacteria that actually, they can also use sulfate and carbon as hydrogen acceptor. And in this case, after oxidation, you normally in our body get water formation because this eventually what happens. Oxygen is the final acceptor of the energy. But in this kind of bacteria, you have sulfuric acid and methane that is actually formed. Because in this case, you don't have oxygen here, but you have sulfate, and here you have carbon instead of oxygen. Yeah? So you obtain completely different molecule. So in the metabolism, what we have that is really good indeed, so it's the coenzyme. Like I told you, otherwise, if we burn the glucose and we just made ATP, we couldn't keep all this ATP. It's just too much. Yeah? It's 5,000 more than what we can keep. It's only 100 grams. Then we use a lot of this coenzyme to transfer energy and make use of this energy only when we needed ATP. Again, this is again to fix the same complex. Remember, this is the reduced form of an X molecule. When it loses hydrogen, this molecule gets oxidated. A coenzyme, and in this case I will show you another one, it is NADH+, then will become NADH plus H+. So this is special, this coenzyme, because it has a positive charge, and since it accepts hydrogen, the positive charge will stay, yeah? Because it's only adding hydrogen to itself, so the charge doesn't change. And then in this case, this is what happens when it will go to the mitochondria. It releases the energy, and then it gets oxidated again. So that basically can be used again in this process. So this is exactly what I showed you before. So try to get how it works with the redox reaction, because we have a lot actually in the metabolism. Okay, that's too much. Again, another type of reaction. So this is, again, what happened, like I told you before. Reduce, oxidated. For something that gets oxidated, the coenzyme, in this case, something else, gets reduced. Yeah? So it's always like this. It's like an X. So one gets oxidated, the other reduced, and the other way around. And these are actually some spoiler reaction you will see in the second hour that belong to the Krebs cycle, where we also have a transfer of energy into coenzyme. Yeah? So to talk about the coenzyme, we have, this is something, again, you don't have to memorize. It's just for you to know. But what is important is that we have NAD that can be also in the phosphorylated form, and in that case, it's called NADP. Yeah? And then it has a positive charge normally on top, so we always talk about NAD plus or NADP plus in the oxidative form, and then in the reduced form it has 2 hydrogen on top. And then we have the riboflavin and the flavin coenzyme, which basically you might know because this is basically vitamin B2, and this is actually the FMN, which is a flavin mononucleotide, and the FAD, which is flavin adenine dinucleotide. So we will mostly encounter this actually. And again, this is the oxidated form. Yeah? In the reduced form, you have a FADH2. This, just to let you know, again, you don't have to memorize that to go from FAD to fully reduce FADH2 is not a single actually reaction, but is a 2-step reaction. But this is just, you don't have to remember. It's just that it happens 1 hydrogen at a time. Yeah? So I just